Gram staining is a technique utilized to identify whether a culture of bacteria cells is gram positive or gram negative. And whether a bacteria is positive or negative all depends on its structure. So if we take a look at gram positive bacteria, if we take a look at the outer coating, the outer shell, the outermost layer is the capsule. So the capsule in red is the outermost layer of a bacteria cell. One step underneath the capsule, we come across the cell wall. And the cell wall in bacteria is made up of peptidoglycan. Now, in plants, we know that the cell wall is made up of cellulose, and in fungi, the cell wall is made up of chitin, but for bacteria, it is peptidoglycan. Now, in gram-positive bacteria, there is a thick peptidoglycan layer, and that's important. Underneath the thick peptidoglycan layer is a single lipid bilayer. So it's a cell membrane. Essentially, we have a cell membrane underneath the cell wall, the plasma membrane. So the outermost layer is the capsule. Then we have the cell wall made up of peptidoglycan, and then we see the plasma membrane. It is important to note that we only have one plasma membrane in the gram-positive bacteria. The gram-negative bacteria will be slightly different. So we have that thick peptidoglycan, and we have a single lipid bilayer. Now in gram-negative bacteria, we still have that capsule as the outermost layer, but we have a thin peptidoglycan layer instead of a thick one. So underneath the capsule, we have that cell wall, and it's going to be a thin peptidoglycan cell wall. But what's important here is that gram-negative bacteria have a double lipid bilayer. That means they have two plasma membranes. The first plasma membrane is located underneath the cell wall, which is the same as we see in gram-positive. But in gram-negative bacteria, we have another plasma membrane right above the cell wall. So essentially, the peptidoglycan cell wall is sandwiched in between two plasma membranes. The plasma membranes are in green. So in gram-positive bacteria, we only had one plasma membrane, but in gram-negative bacteria, we have two plasma membranes. Now, if we take a closer look at the structure of gram-positive and gram-negative, we can see that the gram-positive have their single cell membrane, the gram-negative have two cell membranes, gram-positive have a thick peptidoglycan cell wall, gram-negative have a thin peptidoglycan cell wall, and they both have that capsule. Now, in gram staining, what we do is when we have a culture of bacteria and we want to identify whether it is gram-positive or gram-negative, we start off by adding in crystal violet. Now, crystal violet is purple in color, so if we add in droplets of crystal violet to both gram-negative and gram-positive, the crystal violet will seep through the capsule, the peptidoglycan, and the cell membrane, essentially into the cell. In both cases, it'll seep through. Next, we add in iodine. Now, the iodine simply interacts with the crystal violet. And the iodine as well will seep through the capsule, peptidoglycan, and cell membrane into the cell. So over here, we can see that the iodine and crystal violet have seeped through the capsule, peptidoglycan, and cell membrane. Now, what do we do next? So next, we apply an alcohol wash, and we know that alcohol has dehydrative properties, especially if there is water present. So when we add in the alcohol, so let's denote the alcohol with green. We add in the alcohol in both. Now, since alcohol has dehydrative properties, the capsule is going to be dissolved, and the peptidoglycan layer is going to shrink because alcohol is dehydrative. So this capsule will dissolve and disappear, and this peptidoglycan will shrink in both cases. But in gram-negative, the capsule and cell membrane, they will both dissolve. So the gram-negative will lose its capsule, it'll lose its cell membrane, and its peptidoglycan layer will shrink. And now what's important to note here is that since the gram-positive have a thick peptidoglycan layer, this alcohol is not able to wash away the iodine and crystal violet, because think about it as like a thicker protection coat. So these iodine and crystal violet, they're protected by this thick peptidoglycan, and the alcohol cannot wash them away. But in the gram-negative, the peptidoglycan layer is very thin. So the alcohol is able to wash away the iodine and crystal violet. So to recap, what happens is that since alcohol has dehydrative properties, the capsule in both will dissolve, the cell membrane will dissolve, and the peptidoglycan layers will shrink. And in the gram-negative, the iodine and crystal violet will be washed away because the peptidoglycan layer is very thin. Now over here, we can see that the capsule, cell membrane over here, they've dissolved and our peptidoglycan layers have shrunk. Now in the gram-positive, we still have the iodine and crystal violet, but in the gram-negative, we do not because they are washed away. Now the next thing we do is we add safranin. So safranin is pink in color. So we'll denote it. So it's pink in color, but we only have purple. So this is safranin. And keep in mind that safranin is actually pink, but I only have purple over here. Okay. So the safranin is going to seep through. And what we're going to see is that it's going to interact with the cell membrane for both. So over here, we can see these safranin molecules right over here. Now, after we've added in these safranins, we can make an educated guess. We can say, okay, now if we were to look under a microscope and I saw only pink, would it be gram-positive or gram-negative? Well, we would think that, okay, this one has pink and it has purple. So what color is going to show up? I would say purple is going to show up because purple masks the color of pink, right? So and this one, gram-negative, it only has pink. So if I looked under a microscope and I only saw pink, I would say, okay, this is gram-negative bacteria. But if I looked under the microscope and I saw purple, I would say, okay, it's gram-positive. And keep in mind that it has the pink, it has the safranin, and it has the crystal violet. But we see purple because the purple masks the pink of the safranin. So that's how we identify whether a bacteria cell culture is gram-positive or gram-negative. So to summarize, when we have gram-positive and gram-negative bacteria, when we add in the crystal violet is purple in color, it'll seep through the capsule, the cell wall, and the cell membrane into the cell for both gram-negative and gram-positive bacteria. Same with the iodine. But when we add in the alcohol, the gram-positive bacteria have a thick peptidoglycan layer. So the crystal violet and iodine are, in a sense, protected from the alcohol wash. So they don't get washed away from the gram-positive. But in the gram-negative, since we see the thinner peptidoglycan layer, the alcohol is able to wash away the crystal violet and iodine. Once we add in the safranin is present in both the gram-negative and gram-positive, but since the gram-negative only have pink because the crystal violet gets washed away, the gram-negative appear pink under a microscope, whereas the gram-positive, they appear purple. And it's important to remember that they have both crystal violet and safranin, but the violet masks the pink of the safranin. And that is gram staining.